Hi, my name is Bhoomika, and in this chapter, we're going to deal with the physiology of excretion, mainly nephron function. So, I have already done a lesson on the background data, that is, the structure, the basic structure, and the composition of the excretory system. So, I think before beginning with this one, you should check that one out. Alright, so let's start. I am going to actually start with the forces of filtration, and I have specially done a handwritten tutorial because I think understanding in this way is better. Okay, so I have a glomerular apparatus here. I have the Bowman's capsule drawn. We have the afferent arteriole this side and the efferent arteriole this side, and the blood moves in such a manner. This is the glomerulus, as you can see, and here we've got the Bowman's capsule. Now there are three forces which actually control the glomerular filtration rate. The first force is the hydrostatic pressure. The second force is the oncotic pressure, which is exerted by the proteins in the blood onto the fluids. The third is the net pressure that goes onto the proximal convoluted tubule. So, there is a hydrostatic pressure that develops on the blood, which is present here, which passes to here. When it reaches here, the hydrostatic pressure, it tends to push the filtrates outside towards this side. So this is coming out. Then again, the filtrate which comes out here exerts a hydrostatic pressure which is specific to this filtrate towards the opposite side. Now, what is hydrostatic pressure? Basically, if I asked you to imagine a balloon and I poke a hole in the balloon, so what will happen is the air inside the balloon, it would definitely tend to turn out. So in the same way, a liquid or a solution, it extends some pressure on the walls of the container that it is enclosed in. So as such, the filtrate also does so. So here the blood exerts the pressure onto this side, and the filtrate here exerts the pressure onto that side. So we've got the hydrostatic pressure of the blood here, which is 55 millimeter per mercury, and the hydrostatic pressure of the filtrate is 15 millimeter per mercury. Additional to these, we also have the oncotic pressure, which again I told you is exerted by the proteins. So this is in an opposite direction to the hydrostatic pressure of the blood, and it is about 30 millimeter of mercury. Now in the examination, a number of times a question is asked in which you have to calculate the net pressure. So I have written the equation here. We've got the net pressure is force out minus the force in. So this is the force out and minus the force in. So using the rules of BODMAS, I have done 55 minus this 55 minus 30 plus 15, which is going inside, so minus 45. So the total pressure, the net pressure that is exerted here towards the proximal convoluted tubule is 10 millimeter per mercury. So that was about the forces of filtration. Now let's move on to the Renin-Angiotensin-Aldosterone system. These are all basically hormones. These hormones, they control the excretion rate, the filtration rate. So I'm going to talk about angiotensinogen. Angiotensinogen is secreted by liver, and it is an inactive form. This angiotensinogen, in presence of renin, which is secreted by kidney, converts itself to angiotensin 1. This angiotensin 1 again is also inactive. So this is supposed to be converted into angiotensin 2 by angiotensin converting enzyme, and it is secreted by the lungs. See, first angiotensin 1 is formed, then it freely rotates around in the blood, and when this angiotensin 1 reaches the lungs, the ACE, that is the angiotensin converting enzyme, it comes in contact with it, and it converts the angiotensin 1 into angiotensin 2. Now that this angiotensin 2 is formed, it goes to the adrenal gland, and it stimulates the adrenal gland to secrete aldosterone. This aldosterone goes onto the nephron, and it stimulates the nephron to retain water. This water retention is very important in order to prevent dehydration from the body. Now that water is being retented here, the blood pressure rises. Why does the blood pressure rise? Because the vessels, they are being constricted. So according to the equation of continuity, A1 V1 is equal to A2 V2. So as the diameter of the blood vessels, it gets lower and lower, the blood tends to gush out of the vessels like that. So another hormone it comes into being, it is the vasopressin or the ADH, which is secreted by the pituitary gland, and it helps in the normal dilation of the vessels. So that was about the renin-angiotensin-aldosterone system. Hormonal information is important because all these hormones, they tend to control the filtration rate. All right, so let's move on to the basic physiology. I have drawn a nephron here. So the nephron, it looks anatomically different, but because I had to draw a diagram, I have done it in a very vague format. Here we've got the afferent arteriole and this is the efferent arteriole, and here we've got the glomerulus. So maximum amount of filtration it is done in the glomerulus area. Here water, NaCl, K+, bicarbonate ions, glucose, amino acids, creatinine and urea, they all are filtered at this point right here. Now the blood moves inside and towards the proximal convoluted tubule. You should be thorough by the structure right now, and that is the reason why I have not named or labeled the structures. So this is the proximal convoluted tubule right here, and some secretions takes place at this point, some secretions including uric acids and some organic acids. And also as it moves a bit further, reabsorption takes place, and this reabsorption is of K+, NaCl, amino acids, glucose and bicarbonate ions. Now the filtrate, the rest of the filtrate, it starts moving towards the loop of the Henle. So in the descending loop of Henle, a lot of water is reabsorbed. As you can see, water is being reabsorbed, and in the ascending loop of Henle there is 25% of NaCl absorption. Now the filtrate, it starts moving towards the distal convoluted tubule. Here K+ ions and hydronium ions, they are secreted inside the filtrate, and again NaCl and H2O absorption takes place. Now as you can see, water is being absorbed at every single step, and at some steps NaCl and all the ionic reabsorption is being taken place. So now this filtrate, it starts moving towards the collecting duct. Here again some water, NaCl and urea is absorbed, reabsorbed. Okay, so this is 25%, this is 5%. Here NaCl absorption is about 65%. So that is how an ionic balance it is maintained throughout the body. So now this filtrate, which is the main constituents which are supposed to be retained by the body, have been completely absorbed at this level. So now the rest of the filtrate, it forms the excretion substances. So this excretion substance, it includes creatinine, it includes some amount of water, some ions such as the NaCl, the K+ and the bicarbonate ions. So in this way filtration takes place, and this forms the urine which starts moving here. The main constituents are written here. I have not written all of this in a theoretical format because I thought that a number of books that you may be referring to will have all of this. I just wanted to make it pretty clear with the diagrammatic use. So I hope you all enjoyed this. I hope you all understood the basic concepts. If you want to understand in a different language such as Hindi, I would also be doing this tutorial in Hindi, so you might as well check that out. Thank you so much for watching.